Hello students, welcome back to the channel. Now we will move further in our topic. In the previous video, we have studied blood and its composition, that blood is made up of plasma and formed elements. In this video, we are going to focus the blood groups. So see here, blood of human beings, they differ in certain aspects. Though it appear to be similar because composition is same, but then also they differ. In few certain aspects, they are differing. So various types of grouping of blood has been done. Two such groupings like ABO blood grouping and RH factors, they are widely used all over the world. In this video today, I'll be focusing only on the ABO blood group. Next video, I'll be focusing on RH. So various types are there, but two are very important, ABO blood group and RH blood grouping. So we are focusing on ABO blood grouping. Now these blood grouping, ABO blood grouping, why we are classifying them? So they are based on the presence of or absence of two surface antigens. Antigens are what? They are the chemicals. Definition of antigens. Antigens are the chemicals which can induce immune response. Means they are the one which are the agents which can generate antibodies. So they are present on the surface of RBC. Okay, so they are present on the surface of RBC. And two antigens, which two antigen? A and B. Now, they are present on the surface of RBC. Clear beta? Antigens on surface of RBC. Antibodies are present in plasma. The plasma of different individuals will contain two natural antibodies. The antibodies are what? See the definition. Antibodies are protein which are produced in response to antigens. So this is a definition of antibodies. Clear? Antibodies are what? Antibodies are proteins produced in response to antigen. So up till here, what you have understood, antigens are present on RBC surface and antibodies are present in plasma. What are antibodies? Antibodies are protein. And what are antigens? Are they protein? Are they carbohydrate? Or are they glycoprotein? So for this, we have to see the 12th class. One paragraph is there of the 12th class. Let me show you that paragraph. See beta, this is the paragraph of 12th class, Principle of Inheritance. Topic is codominance. In that it is there. If you see, different types of RBC which determine ABO blood grouping in human beings, and this blood grouping is controlled by the gene I. Plasma membrane of RBC has sugar polymer. It has sugar, okay beta, sugar polymer which protrude from its surface, and the kind of the sugar is controlled by the gene. It means these sugar polymers are the one which are working as an antigen. So blood grouping is the one which has got three alleles. Gene is I, one gene I, okay? This gene is I, blood group is controlled by gene I, and it has three alleles. What are the three alleles? IA, IB, and small I. So you can see these are the three alleles. Come down in the diagram. Allele IA will make carbohydrate A, IB will make B carbohydrate, and I small I will not be able to make any carbohydrate. So it means that is why in our 11th class they are telling us that there are two natural occurring antigens on the surface. So only two. Alleles are three, but antigens are only two. IA and IB, they produce a slightly different form of sugar. Allele small I does not produce any sugar. Is it clear, beta? So now if you want to see, check beta. If suppose the person genotype is this condition or this condition, only A sugar will be there. We say blood group is A. Phenotype is actually a blood group. If this condition is there, only B sugar will be there, blood group will be B. If both different allele, why? Because we are diploid, we will have two alleles. Either allele can be same or allele can be different. If you see, this is heterozygous. This is heterozygous. Even this is also heterozygous. In first two cases, if you see heterozygous, only one was able to express because dominant recessive relationship is working. But in this case, both are dominant. That is why two types of sugar are made. A sugar is made, B sugar is also made. Clear? And in the small I, if you see there is no sugar, O means no sugar, zero sugar. So I hope so now this you are getting an idea. So from the explanation what I have given you right now from the 12th class NCERT, what are antigens? Please note down, antigens are carbohydrate, because this is a very common question they ask. Okay, beta, antigens are carbohydrate. So if you see this distribution of antigen and antibodies in the four blood group, so you know very well. Blood group is A. Right now we have seen the antigen will be A only. Blood group is B. Antigen will be B only. Blood group is AB. Why? Because both antigens are present. Both types of sugars are present. Blood group is O because there is no antigen. And you know antibodies are present in plasma. If blood group is A, you will not have same antibody. Antibody will be B. Blood group is B. Antibody will be A. AB blood group will not have any antibody. Why? Because it has both antigens. And O blood group will have both antibodies. So distribution of antigens and antibody in the four types of blood groups, I think so you have understood up till here. Now, during blood transfusion, any blood cannot be used. We have to be carefully match the donor and the recipient, so that the clumping of the RBC should not occur. And clumping will, now please remember, we have to, when you are doing blood transfusion, see during blood transfusion, any blood cannot be used. The blood of donor has to be carefully matched with the blood of recipient before any blood transfusion. Why? We don't want antigen antibody interaction. So see carefully, donor, if any person who is donor, antigen is important. We have to check antigens of the donor. And we have to check antibodies of recipient. They should not be same. Suppose, let us assume one person is there, person blood group is A. Okay, the person blood group is A. If A blood group, it means antigen is A and antibody is B. This much is clear now? Now, who can give the blood? Let us assume this person is recipient. This person is recipient. So we have to see the donor antigen. I told you, donor antigen is important. We have to check carefully that the recipient antibody is B. So I should not give B antigen. The person should not get B antigen. And blood group B is having B antigen, so I should not give blood group B. So first of all, blood group B cannot be given. Why? Because B will have antigen B. And antigen antibody, same clumping will occur. I cannot give blood group AB also. Why? Because AB blood group is having antigen B. Because it has both antigens, it is having antigen B. So again, clumping will occur. So these two blood group I cannot give. Which blood group I can give? I can give the person who can give the blood group. That is, A blood group can give, same blood group. Another one who can give is O blood group. Why? Because O blood group does not have any antigen. Please remember, donor antigen is important. We have to check donor antigen should not match with the recipient antibodies. They should not be same type. If they become same type, clumping will occur. Understood? Now, if suppose I ask you, blood group B is having antibody A. It means antigen A should not come. Antigen A matching should not be done. So antigen A is present in blood group A, it is present in blood group AB. So these two persons cannot give. So he can receive blood from whom? He can receive the blood from the B and he can receive blood from the O. So if you see in this table, from this you can check O is the one who can give it to everyone. O is the one who can give it to everyone. Why? Because O does not have any antigen. And AB blood group is the one which can take from everyone. Is it clear? It can take from A also, it can take from B also, it can take from O, AB. It means AB blood group is universal recipient. Why it is universal recipient? Because antibodies are nil here. So it is evident that O blood group can be donated to person with any other blood group. That is why O is called as universal donor. And person with AB blood group can accept blood from person with AB plus as well as other. We say such persons are universal recipient. So I hope so in this video, the topic might be clear to you. There are different types of blood groups are there, but two are widely used, that is ABO and RH. ABO blood group is controlled by a single gene I with 3 alleles, out of which 2 alleles are able to make sugar on the surface of RBC working as an antigen. 2 naturally occurring antibodies present in the plasma. Which 2 naturally occurring antibodies? That is antibody A, antibody B. So person blood group will be determined by the antigens. If antigen is A, sugar A, blood group is A. And antibody will be opposite, that is antibody B will be present in the plasma. Based on this, 4 types of blood groups are there: A, B, AB, and O. In which we find O is universal donor because donor blood group does not have any antigen. O is universal donor because it does not have any antigen. So it will not be able to generate any antibodies. AB is said to be universal recipient because it does not have any antibodies to destroy the antigens. So I hope so in this video, the topic might be easy for you. Thanks for watching.